First, we're going to talk about the nitrogen cycle. Nitrogen is converted into nitrite by bacteria, which is then converted into nitrate by nitrobacteria, and this is when a plant is able to use it. When a consumer, like a bear, it comes and eats a fruit, it first eats it, then the organism, like the bear, would die, and it's released into the atmosphere by bacteria, releasing it into ammonia, which is called ammonification. Then anaerobic bacteria convert ammonia into nitrogen, and this is a process called denitrification. Nitrogen is important because it's used in bases for DNA like guanine, thymine, adenine, and cytosine. Now we're going to talk about mitosis. First starts with prophase. Genetic material is duplicated inside the nucleus, and the spindle fibers start to form. Metaphase. The central mirrors of the chromosomes line up in the middle, and spindles connect to each of the central mirror of each chromosome to the two poles of the spindle. Anaphase. The chromosomes separate and move along the opposite ends of the cell. Telophase. Chromosomes line up and begin to spread out into tangled chromatin. And then cytokinesis, and this is where a cell plate forms in the middle of the original cell and divided into two nuclei. The plate develops into a cell membrane, separate the two daughter cells, and the cell wall develops between a membrane. This is special inside the plant cell. Then there's meiosis. This is basically where parents are able to replicate their genes and then connect them to others of their certain species. It first starts with meiosis 1, where the cell first duplicates all of its chromosomes. Then prophase 1, each replicated chromosome pairs up with the corresponding homologous chromosome. Then they form tetrads and then cross over parts of their chromatid. Metaphase, spindles form and attaches to the tetrads. The paired up homologous chromosomes line up in the middle. Then anaphase 1, the spindles pull the chromosomes toward the opposite end of the cell, separating the tetrads. Telophase 1, the nuclear membrane forms around the cluster of chromosomes, and then cytokinesis forms two new cells. Then it goes to mitosis 2, which is still part of meiosis. It goes to prophase 2, where the chromosomes become visible. They do not form tetrads because there are no homologous chromosomes next to each other. Then there's metaphase 2, where the chromosomes line up in the middle. Then anaphase 2, the chromosomes split in half, separating the chromatids. And telophase 2, a nuclear membrane forms around a group of chromatids. Then it goes to cytokinesis, where this forms four new cells called haploid daughter cells. Then we start heading to DNA. The reason why DNA is so important is because it holds the genetic code for each individual animal. Now, when two species mix their genetic material together, they're able to create an offspring with variation of their traits. Next, we're going to talk about the process of transcription. Segments of DNA serve as templates to produce complementary RNA molecules. This happens in the nucleus. We're going to talk about translation, which occurs in a ribosome. Ribosomes use a sequence of codons and RNA to assemble amino acids to make polypeptide chains, which are able to make amino acids, which are basically proteins. Mutation happens in the DNA structure whenever UV lights or certain chemicals are able to disrupt the pattern, which will cause a unique trait to happen in the plant. For example, if corn were able to grow taller, and this would be great, it's because it would be able to get more sunlight, but it'll also be bad for wind and crows to come along and rip it out of the ground.